Welcome everyone. Today we are going to learn about mobility of membrane proteins. But before I jump to the main topic, I want to share one information that in 1970, scientists proposed one model called fluid mosaic model of the plasma membrane. Now the term fluid is because of the mobility of the lipids and proteins in the plasma membrane. So the plasma membranes are made up of protein and lipid. So both these two molecules are actually mobile in the plasma membrane. I have already discussed about the mobility of lipid molecules in the plasma membrane in my previous video, and in that video I have mentioned that these lipid molecules generally perform four different movements. One is called lateral diffusion, flexion, rotation, and flip-flop. Now flip-flop is actually migration of lipid molecules from one layer to the other layer of the plasma membrane or vice versa. So here, as because this plasma membrane is bilayer, lipid bilayer, so lipid molecules can actually migrate from one layer to the other. So if you have not watched this video, you can go and watch this video also. Now let's talk about membrane mobility of membrane proteins. At first, we have to understand that membrane proteins have never been observed to perform flip-flop. In case of lipid molecules like phosphoglycerides, sphingolipids, they rarely perform flip-flop, but one exception is that cholesterol, it can flip-flop rapidly. But in case of membrane proteins, they have never been observed to flip-flop. So one point we have to remember, no flip-flop of performance or mobility occurs in case of membrane proteins, but they can show rotational diffusion just like the lipid molecules and lateral diffusion. Now, most GPI anchored proteins. Now, what is GPI anchored proteins? I have already made a separate video on membrane proteins. So in that video, I have mentioned that there are three major categories of lipid protein molecules present. One is called integral membrane proteins or transmembrane protein, and it is called lipid anchored protein, and third is peripheral protein. Now, GPI anchored proteins actually falls under the lipid anchored protein because here phosphatidyl inositol, one phosphoglyceride, is actually present in this protein molecule. So proteins are actually attached with this phosphoglyceride. So most GPI anchored proteins diffuse freely, but a fraction of these proteins have restricted mobility. So the diffusion is not all the time it occurs, but it has a restrictions in their mobility. Now, some transmembrane proteins also diffuse freely, but a fraction may become trapped or immobilized at any time. Now, examples like cell adhesion proteins, cadherin, and nutrient receptor like transferrin receptor. This transferrin receptor actually transport iron into the cell. They diffuse intermittently. That means they alternate between free diffusion and temporary trapping for 3 to 30 seconds. So they actually alternate between these two states of the mobility in local domains, because plasma membrane contains some domains that are protein rich and another is lipid rich. So they actually move or show mobility in local domains measuring less than 0.5 micrometer in diameter. Now, this fluorescence recovery after photobleaching is actually a method by which we can actually measure whether a protein or lipid molecules is laterally diffused, actually perform lateral diffusion or not. So lateral diffusion of membrane proteins can be measured by using this method. I have already made separate video on FRAP or fluorescence recovery after photobleaching this method. Now I will also talk about these methods in this video. So what happens here, we actually take a cell and actually dye or actually stain the cells with antibody attached with fluorescent dye. So fluorescent dye actually fluoresces and gives a color. So let's suppose the antibody that is attached with the fluorescent dye gives a green color. So the cells will stain like green. So it will fluoresces the green color. So it is called fluorescence. Now what happens, we actually photo bleach a small area by laser beam. So with the help of laser beam, a small area is actually bleached, so we call bleached area, and in that bleached area no fluorescence is present in the protein molecules. Okay, so proteins are targeted with a particular antibody. This antibody attached with a fluorescence dye. Now this fluorescence dye is actually bleached with the help of laser beam. So one segment of the membrane is actually bleached with laser beam. Now what happens if we give some time to the cell? If there is a recovery occurs of the fluorescence recovery occur, that means that proves that some unbleached proteins actually move into the bleached area, or the bleached proteins actually move out of the bleached area. So two things can occur. So recovery, if there is recovery, that means there is lateral diffusion, and if there is no recovery, that means the proteins are not performing lateral diffusion. But scientists, what they found, that after bleaching, the proteins actually, the fluorescence actually recover. Basically the recovery of fluorescence indicates that the protein is actually mobile. So this index, this x y axis actually shows the fluorescence in bleached area. At first, before the bleaching, the fluorescence is in this level, okay, you see, in this level. But after bleaching, the fluorescence actually drop suddenly. Now if we give time, then the proteins that are actually present in outside the bleached area, that contains the fluorescent dye, can move into the bleached area, or the bleached proteins can move out of this bleached area, so they will be mixed, and this fluorescence will come again. So it is recovery. But as you can see, the recovery is not complete, and it indicates a substantial fraction of protein is actually immobile. Okay, now let's see how it looks. So as you can see, this is the conditions before photo bleaching, and after that this area is actually shows no fluorescence at all. But after 1 second, some fluorescence is actually coming, so that shows that some proteins are actually mobile. But after 10 seconds, it actually not recovers completely, as because uh, the before conditions of the photo bleaching shows this much of density of the fluorescence, and here is not complete recovery occurs. So this technique called fluorescence recovery after photo bleaching. So we actually fluoresce the cells, then perform photo bleaching, and then we actually observe the recovery. If there is no recovery, that means no lateral diffusion. If there is recovery, that means there is lateral diffusions. So by this technique, we can actually identify the fluorescence lateral diffusion. Now, the first experiments to demonstrate that membrane proteins could move means here lateral diffusion, can perform lateral diffusion within the plane of membrane, okay, utilized cell fusion. So this experiment actually does not depend on the fluorescence recovery after photobleaching, or in short FRAP. But in this case, the first experiment that shows that proteins are actually mobile is performed by Larry Fry and Michael Edidin of Johns Hopkins University in 1970. And what they did, they actually fused cells. So this is the original paper of the published by Fry and Edidin. The title of the paper is the rapid intermixing of cell surface antigens. Now antigens is actually protein, so cell surface proteins after formation of mouse human heterokaryons. Now they actually fuse two different species cells, mouse and human. So when they mix, we call heterokaryons. Now what they did, they actually take the mice cells and they stain these mice cells with antibody that attached with a green fluorescent dye, and in case of humans, they actually stain the human cells with a antibody particular that can bind with the human antigen or human protein, plasma membrane protein, with a antibody attached with a red dye. So when they mix these two or fuse these two cells, they actually found that the two protein of the two cells actually present in two hemispheres, because this is the just fusion after the fusion of the two cells. But they observed that after 40 minutes of intubations of these two cells, the two protein molecules, that means the green and the red fluorescence, actually mixed. And this is the schematic diagram of the same experiment. As you can see, this is the human cells that fluoresces red color. These are the proteins that is actually attached with the antibody of red fluorescence dye, and this is the mouse with green fluorescent dye. When fusion occurs, different hemisphere, but after 40 minutes of incubations, the green and red proteins actually mix, and that shows that there is lateral diffusion. So not only by FRAP experiment, but by this fusion experiment also, these two scientists actually discovered, Larry Fry and Michael Adedin, discovered that these protein molecules are actually mobile in nature. Now, some there are some drawbacks of the FRAP experiments, and these are the it monitors this FRAP experiment can monitors the movement of large populations of molecules, in this case these protein molecules, in a relatively large area of membrane that is 1 micrometer in diameter. But one cannot follow individual protein molecules. So here a group of species of proteins are actually stained with antibody, and we follow the whole proteins, not a single individual proteins. So if a protein, now in this experiment in case of FRAP, if a protein failed to migrate into the bleached area, for example, one cannot tell whether the molecule is truly immobile or just restricted in its movement to a very small regions of the membrane, because I have mentioned previously that proteins are actually mobile in a very restricted area that is about 0.5 micrometer in diameter. But here in case of FRAP experiments, they are actually observing 1 micrometer diameter. So maybe one protein can be restricted to a small domain that is protein rich in segment, so that domain, but we cannot confirm that whether this protein is truly immobile or just restricted in its movement. Now, one method to actually overcome this problem is called single particle tracking technique. Now, single particle means here a single protein is actually stained with this antibody, and the antibody can attach with a gold particle or a bead. So we can actually track the single protein by video microscopy. So this is called single particle tracking. This technique can overcome the drawbacks of FRAP. As you can see, this is the proteins that is attached with antibody that attached with the bead, and you can observe the bead moving around the membrane. So one can record the diffusion path of a single membrane protein molecule over time, and it actually overcome the problems that when we actually fail to conclude that whether the protein is actually really immobile or just restricted. Okay. Now let's talk about the movements that are performed by proteins. So here six types of basically protein movements can be seen. One is the first is A, that shows largest fraction of membrane proteins move randomly. So there is no directions; they just randomly move around the plane on the plane of the plasma membrane. So this is called lateral diffusion. So largest fraction of membrane proteins move randomly. This is the example of A. Now some membrane proteins fail to move and are considered to be immobilized, like the B proteins, they are immobilized. Now in case of some proteins, in some cases a protein is found to move in a highly directed non-random manner, like the A protein. So in case of C, this type of proteins move in a particular directions, and now this direction is actually provided by two molecules like actin and myosin. Now movement is in case of D, the protein D, actually the movement is restricted by other integral membrane proteins. So this situation can happen also for some proteins. Now for E, movement is restricted by fences. Now fences are made up of membrane skeleton, cytoskeleton, that are present beneath the plasma membrane. As you can see, these are the cytoskeleton, membrane cytoskeleton. So movement is restricted by fences formed by proteins of the membrane skeleton, but it can hop into adjacent compartments through transient opening of the fence. And in case of F, the movement is restricted by extracellular materials that are present outside the cells.